Students, this is Dr. Maruti Jairam Dhanodi working as assistant professor at Bharati Vidyapit's Dr. Patangrao Kadam Mahavidyalaya Sangli. In previous lectures, we were discussing microbial growth, and in that point, we have discussed sub points like what are the physical requirements for the microbial growth, and physical requirements like pH, temperature, osmotic pressure, and also we have discussed different chemical requirements for the microbial growth. And in chemical requirements we have discussed what is the carbon source, what is the requirement of nitrogen source, sulfur, phosphorus. These are the requirements of carbon, nitrogen, sulfur, phosphorus, which are included in a chemical requirements. And then finally we have discussed what are the different medias and what is meant by culture media, and what are the different types of medias like differential media, then chemically defined media, then complex media. So based on the requirement of nutrients, different organisms require different media, and whenever we provide a particular nutrients in a media, the specific type of microorganisms can be grown, and these microorganisms can be kept as a pure culture. So also we have discussed what is meant by pure culture and how we can obtain a pure culture. A pure culture is nothing but the culture in which single microbial species is present. So we know that most of the clinical and environmental specimens contains different organisms. So n number of organisms are present at clinical and environmental samples. So to isolate a single organism, what we have to do, we have to provide a specific medium and a specific condition. When we provide a specific medium, a specific bacteria can be grown. So how we can specify or how we can purify a bacteria from a mixed culture in a environmental samples? So to obtain a pure culture, we have to use a particular techniques, and by using these techniques we can purify a culture from a mixed environment. The most common method for isolation of pure culture is strict plate method, in which a sterile loop is inserted inside into the sample, and that loop is strict on a plate which contains media in a specific pattern to obtain an individual colony. So when we have a medium kept inside into the plate, that medium must be sterile. When we have a sample, we can insert a nichrome wire loop inside into that sample, and we can stick that sample on a media in a specific pattern. So after incubation of this plate which contains solid media, after 1 or 2 days there will be formation of colony on the liquid, on the solid media, and that colony is nothing but a group of descendants of original cell. A colony is nothing but a colony contains large number of organisms which are from a similar background, or from a single cell there is a formation of large number of cells which forms the colony on a solid media. So culture, we can obtain that culture can be called as a pure culture when we took a pure isolated colony, and when we stick that pure culture inside into another plate, that plate is containing a culture in which single type of cell is present, so that we can call it as a pure culture. So when we have a plate in which single type of organism is present, then that plate can be called as a plate having a pure culture. Okay, so growth of bacterial culture, how we can do? The bacterial divisions occurs mainly by binary fission. A few bacterial species reproduce by budding. So what is generation time? The time required by the cell for division, okay, and produce its population doubled. Okay, so that is called as a generation time, the time required for a cell to divide and produce its population. to divide its population, the time required is called as a generation time. So generation time varies considerably. E. coli divides every 20 minutes. Most of the bacteria can divide every 1 to 3 hours. Some bacteria requires 24 hours for the division. So different bacteria require different generation time. E. coli is the fastest one, which required 20 minutes for to double its number. So generation time of E. coli is fast. Within 20 minutes from a single cell, 2 E. coli cells can be produced. Then this is the bacterial growth by binary fission. We can imagine the single cell which contains plasma membrane and cell wall and internal cytoplasm with genetic material. Whenever cell is grown, which will increase in size, in which there is a large amount of cytoplasm is produced, as well as there is a duplication of genetic material. Whether it is DNA, the duplication of genetic material is happen along with the duplication of cytoplasm. When cell attains its proper size, cell is going to divided into two cells. So in left side you can have a picture in which from a single cell, by the process of binary fission, the two cells are formed. So microorganisms mainly divide by the binary fission. And how does division is occurring? Division is occurring whenever cell is fully grown, there is an increase in cytoplasm, increase in cell membrane, increase in genetic material, then cell is ready to divide. After attaining optimum size, cell is ready to divide and single cell is divided into two cells. Then this is all about the bacterial growth, generation time and pure culture. Then we will see how the microbial growth is happening, the growth of culture, in which there are different stages. The logarithmic representation of bacterial growth curve is possible. We can express the number of cell in a bacterial generations as 2 raised to 1, 2 raised to 2, 2 raised to 3. In this manner we can represent the bacterial generation time. Okay, bacterial generations, where n is the number of doubling that have occurred. So when 2 raised to 1, the doubling occurs once. When 2 raised to 2, the doubling occurs 2 times. So we can represent a generations as 2 raised to n. So number of bacteria we can count based on the generations. For example, if initially E. coli is a single cell, then after 20 minutes it will produce 2 cells. Then after 40 minutes it will produce 4 cells. After 60 minutes it will produce 8 cells. So in this way we can predict what is the actual number of cells in a particular colony based on the generations as 2 raised to n. So there are different phases of growth. First phase is the lag phase. So bacterial growth curve, when bacteria are inoculated inside into the liquid media, we can plot a number of cells in a population over time. So we know that with respect to time, the bacterial number will goes on increasing. So as time passes, the number of cell is going to increase. So we can plot a number of cell on x-axis and we can plot a time on y-axis. So based on that graph we can represent the number of bacteria with respect to time. So there are 4 different phases of growth. First phase is a lag phase, in which the bacteria, whenever kept inside into the media, the bacteria firstly in a lag phase enter inside into the lag phase. So before lag phase there is a zero phase or lag zero, which is also called as a resting phase, in which bacteria is not metabolically active. But in lag phase, Also, bacteria is not that much metabolically active. What are the activities needed for the bacteria's survival is going on inside into the bacteria, but bacteria is not in a mood to grow and divide. Therefore, it is called as a lag phase, and the period of adjustment to the new conditions. So, in a lag phase, whenever bacteria is kept inside into the new media, there is a different conditions. So, bacteria will start to adapt to that conditions. For example, when we provided a medium to the bacteria, the medium may contain a specific pH, the medium may contain a specific nutrients, so that bacteria whenever kept inside into that medium, bacteria try to adjust at that conditions of different pH, temperature and nutrients. So the lag phase is the period in which bacteria try to adjust to the new conditions. There is a little or no cell division occurs in a lag phase. The population size doesn't increase. The phase of intense metabolic activity in which individual organisms grow in size, then mainly last for 1 hour to several days. So whenever we introduce any bacteria inside into the medium, the lag phase initiated, and lag phase may last for few hours to several days. And mainly you have to keep in mind that in a lag phase, bacteria or organism try to adjust to the conditions present inside into the medium. Then second phase, log phase. In log phase, cell begins to divide and generation time reaches a constant minimum. So in log phase, we can say that it is also called as exponential growth phase, in which cell start to divide and increase their progeny. Okay, so in log phase, there is a division of cell is occurring. The generation time reaches a constant minimum. The period of most rapid growth, that is called as log phase. The number of cell is produced is equal to number of cell is dying. The cells are highest metabolically active. The cells are most susceptible to adverse environmental factors at this stage, like radiation and antibiotic. So in log phase, cell is active in nature. Actively it is going to increase their progeny. Then cell is also sensitive to different things like radiation and antibiotics. It is also susceptible to the environmental factors at this stage, like log phase. So do you understand what is lag phase and what is log phase? So in next lecture we will study another different phases of bacterial growth curve. Thank you.